And erythema migrans is pretty much like a visual site of the Borrelia burgdorferi spreading under the skin. They've actually, like, even when you can't find the antibodies for it in the serum, you can actually find the bacteria in the outer ring of the erythema migrans. And then after it leaves the erythema migrans, it either pools in the joint, heart, or brain. And so if it pools in the joints, it's usually the knees, and that leads to arthritis. This especially is very noticeable in children. And then when it spreads to the heart, it usually results in heart blockage. And when it spreads to the brain, it can lead to encephaly or other nervous system problems. And these conditions are usually caused by either the outer surface toxins being next to those tissues for too long, or it's because of how long it takes the immune system to get rid of those pathogens, and having long-term inflammation causes the damage. And then this is just a list of the symptoms that are released by the CDC. The first two bullets are symptoms that usually appear within the first month of infection, and then the last four are ones that happen after that. And as you can see here, the erythema migrans is the only early symptom that's unique to a tick-borne illness, whereas the rest of the symptoms you could just attribute to a cold. And erythema migrans doesn't appear in 100% of cases, but it does appear in about 80%.
All right, so I'm going to talk about the epidemiology and social impact of the disease. So for a little bit of history, researchers believe that Lyme disease was present in nature long before the first case was seen in humans. The first report of the erythema migrans rash was in Sweden in 1909, and it was reported several more times across Europe in the next several decades. In 1977, a group of children in Lyme, Connecticut had similar symptoms to each other, including childhood arthritis, and that is now considered the first endemic of Lyme disease in the U.S. The bacteria Borrelia burgdorferi was isolated as the cause of Lyme disease in 1982, and around the same time it was discovered that ticks in the genus Ixodes are the primary vector of the disease to humans. For the past 4 decades, the number of cases has steadily increased in the northeastern United States. So ticks have 3 main stages of life: larva, nymph, and adult, and they have to attach to an animal for a blood meal once per stage. It's likely that ticks can't pass the bacteria between generations, so a tick must eat from an infected host animal to get the disease. If a mammal can pass the disease to a tick, they must have been infected by another tick and also be competent or able to take the bacterial DNA into their own genome. Some mammals, like horses and deer, are not competent and can't spread the bacteria between ticks. Ticks are often infected in the larval stage of life and often infect humans during their second feeding during the nymph stage. Humans and domestic animals aren't part of the natural and zootic cycle of Lyme disease, which means that the disease is present in nature without human hosts. Lyme disease is spread through competent small animals, and the white-footed mouse is the most common reservoir host. Ticks in the genus Ixodes spread the disease to humans in the U.S., and the deer tick is the most common species in the Northeast. They usually attach to humans during the nymph stage of life and sometimes the adult stage, but adult ticks are more likely to attach to large animals like deer and horses. Ticks have to stay attached for roughly 24 hours to transmit the bacteria, but people rarely know for sure how long the tick has been attached. So if someone knows that they were bitten by an infected tick, they will usually start treatment. Nymph ticks are super, super small and hard to notice, so it's important to shower after being outside and inspect all your little nooks and crannies, especially those near the seams of clothing. Right now, it doesn't seem likely that the bacteria can spread between humans or from animal to human, but if your pets go into the woods, they can bring the ticks in closer proximity to their owners. And it's actually looking like birds that migrate to different parts of the country at different times of the year are contributing to the spread of the disease across the U.S. So because the disease's in zootic cycle doesn't involve humans, no medical properties make a human more or less at risk for being bitten by an infected tick. Risk of exposure to infected ticks is mostly linked to the environment someone lives in, the time of year, and the measures they do or do not take to prevent tick bites. The Northeast and Midwest have a much higher occurrence of the disease than the rest of the country. Ticks aren't active during the winter, so the risk of exposure is higher during warmer months, and immunocompromised patients are at higher risk of severe infections and prolonged effects because both the innate and adaptive immune systems play major roles in fighting off the infection. Also, if someone doesn't seek medical treatment, if they think or know they were bitten by an infected tick, they won't receive initial antibiotics, and they're more likely to have prolonged symptoms. So only 300,000 cases are reported to the CDC every year, but they estimate that there are closer to 300,000 cases. Many cases go unreported because local governments don't always report cases to the state health department. The estimate of 300,000 was calculated by averaging the number of positive lab results for Lyme disease and the number of insurance claims for the disease made every year. The physical effects of the disease can lead to some major social changes. The joint nerve pain can make it hard to participate in sports or recreational activities, and it can be hard to attend school or complete schoolwork. The dramatic change in lifestyle can also lead to negative effects on mental health. So for treatments, well, ticks are everywhere, so you got to be careful. So prevention is key. This is a really big point we want to drive home. It's good to stay away from deep woods. Ticks can't fly, so they'll sit on the end of a twig or leaf and just wait for something to brush up against them so they can grab onto it. I've even heard of people noticing a tick sitting on the end of a long blade of grass, and its front legs are just stretched out waiting for a host to get close enough to attach to it. So it's important to check your body, check your pets and outdoor equipment after being outside, and wear protective clothing when going out, and use bug spray that contains DEET.
Okay, so for current treatments, Lyme cannot be serologically diagnosed before 2 weeks of infection. And when it is able to be serologically tested, you have to use both an ELISA and an IgM or IgG immunoblot so that you have 2 positives to make sure that it is in the bloodstream, because the bacteria is really good at hiding. And so then when it is definitively diagnosed, it is usually treated with doxycycline because it has the smallest dosage required in milligrams, but it can also be treated with amoxicillin and cefuroxime acetyl, which is really fun to pronounce. And it is often kids are often not prescribed doxycycline, and instead they're given amoxicillin because it's less likely to initiate an allergic response. And in the 90s, they actually did create a vaccine for Lyme disease. However, physicians didn't really like it because it was very expensive and required a lot of booster shots, and they figured it would be a lot easier just to prescribe antibiotics whenever they figured somebody had it. And then one way that physicians have tried to help diagnose Lyme disease before it gets to the 2-week point is through tick testing services like Tick Report, which is sponsored by UMass Amherst. And what you do is you find the tick on your body, you take it off, you bag it, and you send it to their lab. And as you can see here, they can find out the species, the sex, about how much it's fed on a person, and they test it for all strains of Borrelia. Here it is positive for Lyme disease. This is actually a tick that we found on my dad a little bit ago. It's not great, but with this, the patient also says for about how long they think the tick was attached. You usually go by the last time that you were outside for a long amount of time, and then this report can be sent to a physician, and the physician will then make a better decision in either prescribing antibiotics right away or waiting, because there has been incredibly good data that has shown that if you take antibiotics within 72 hours or within a week of a infected tick bite, it almost guarantees that you will not have later symptoms, and it can be eradicated much earlier than it normally is.
So like Megan said, a vaccine was developed years ago, but it was taken off the market because it was expensive, required a lot of booster shots, and Lyme disease wasn't very prevalent at the time. Prescribing antibiotics seemed like a better form of treatment. Now the need is greater because of antibiotic resistance. A vaccine called VLA15 is being developed to attack Lyme disease, and it's showing a lot of promise. The vaccine is being developed by a French company and targets 6 different strains that are mostly seen in North America and Europe. It's considered a multivalent protein subunit vaccine that trains the body to target the outer surface protein A on the bacteria. It's shown positive results in early trials because the surface protein targeted is one of the most prevalent surface proteins on the bacteria when it's isolated from infected ticks. The vaccine is set to enter phase 2 trials soon, and the first results from the trial should be available within a few months. For medications, a group of researchers at Stanford discovered a compound that they hope will become an oral drug to treat the initial and prolonged effects of Lyme disease. Aslocillin has proved able to kill drug-resistant B. burgdorferi bacteria in the lab and in animal trials, so the next step is a clinical trial to test its safety and effectiveness in humans. The drug has the potential of curing the lifelong symptoms of Lyme disease. I found this information in an article that was published only 2 months ago, and the information from the vaccine article was published, I think, on April 30th. So the medication and the vaccine seem to be potential breakthroughs in the treatment of Lyme disease. And then we have our 2 questions and citations. And that's it.
All right. A couple questions for Megan and Rachel.
Did you guys say how long, like once you get bitten by a tick and you're diagnosed with Lyme disease, that you'll have it?
I think typically this like major symptoms aside from the erythema migrans rash take about at least a month to manifest, and it can take up to 4 months for significant arthritis to manifest.
And those effects, because of drug-resistant bacteria, have the potential of lasting a lifetime.
Oh, okay. Ooh.
Did either of you happen to see, because I know you said it won't spread Lyme disease, the ticks won't spread Lyme disease to deer and to horses. Did you see anything about cattle? Because, cattle are usually out in the fields all day, a lot of times in a bunch of tall grass. And so, can cattle get Lyme disease? And is that or do you see anything about them spreading it?
I actually didn't see anything about cattle spreading it. I would assume that they're in the same realm of like deer and horses because they're a large animal, and usually competent animals that can spread the disease between ticks are small rodents.
Okay. One more question?
Does Lyme disease have like a different course in children, or is it pretty similar, like the symptoms and the time frame and all that?
To my understanding, it's about the same. Maybe the only difference might be like the size of the body. It might spread faster due to that, but all of the articles that I read seem to suggest that it affected adults the same way it would affect kids.
All right. right, ladies, good job. Did you see the Dropbox on Moodle for your presentation?
Yeah, we'll
So,
drop it in there.
cool. And I'm recording these so they'll be up and live and active as you guys study too. So good job. I like it. Who's gonna go next?
Me and Amanda will go.
All right, let's do it.
All right, can y'all see my screen? Great. So hi, I'm Amanda, and my partner Gloria and I did our presentation on Candidial vulvovaginitis, more commonly known as a vaginal yeast infection. So to start it off, we're going to be talking about the epidemiology and social impact. And to really understand that, we're going to start with the history of the vaginal yeast infection. And from a medicinal standpoint, this really began being identified in the mid 1800s by two scientists, their names being J.H. Bennett and Lagenbach. And they were researching thrush, which is the white discharge that's produced from a candida infection, which we'll talk about later is the bacteria that actually causes a yeast infection. And they didn't know that that was the cause at the time. They just saw this production of thrush in multiple areas in the body. And so they were attempting to isolate its origin. And in 1846, a scientist named Berg kind of built off of this research. And he concluded that the thrush that was seen was caused by a dimorphic fungus, Candida. And shortly after Candida was really identified as the origin of this, it was able to be pinned down as the origin of Candidial vulvovaginitis. And those medical diagnoses could really begin, but we're certain that women have been experiencing these and suffering from these all the way back to ancient times. And we kind of see that through the development of different herbal remedies and different just like at-home remedies such as yogurt, garlic, and coconut oil. And as you'll see from this little graphic that I have over on the side, some people are really trying to go back to that origin and take a non-medicinal approach. So how do vaginal yeast infections spread? Now, this isn't something that can pass from human to human, like a disease necessarily, but it's really caused by an imbalance of yeast and bacteria in the vagina's natural microbiome. The bacteria that causes this, Candida albicans, is already naturally present in the vagina's natural microbiome and can only really become problematic when that proportion of healthy bacteria to yeast becomes imbalanced. And this can be caused by using antibiotics, having uncontrolled diabetes, being immunocompromised, or having high levels of estrogen. So who is at risk particularly for vaginal yeast infections? And the groups that I'm about to talk about are groups that have a particularly high susceptibility, but it's also worth noting that just any woman of childbearing age is susceptible to a yeast infection. So the particularly high risk categories would be women who are pregnant, specifically because of the different hormone imbalances that they could potentially have. And adding on to that, women who are taking hormone supplements or birth control, again, which can raise their levels of estrogen. Women who are taking antibiotics, and that can be really problematic because antibiotics can kill off a lot of the healthy bacteria that women have in their vaginas, and so it can really give that Candida albicans a chance to overgrow. Uncontrolled diabetes or high blood sugar levels can also be problematic because it really leads to a higher percentage of urine or of sugar in the urine, and that can give Candida albicans the food that it really needs to overgrow and cause this infection. Additionally, patients or women who are immunosuppressed or have an impaired immune system, and a lot of that can be caused by just different treatments they're receiving, potentially if they are taking antibiotics, and it can just take a longer time for the infection to go away because of their compromised immune system. And additionally, women who are living in environments where Candida albicans can really thrive, and those specifically are tropical environments, locations with low access to hygiene resources or potentially even hygienic education. And it can become really problematic when a woman is living in an area where multiple of these factors are at play, so potentially a tropical environment where there is low sanitation resources. So what are the additional effects of vaginal yeast infections? So outside of just the traditional medical physical symptoms, women who are suffering from chronic yeast infections, which is having, I think, more than 3 yeast infections within a period of 6 months, are also have a higher susceptibility to developing depression, anxiety, low self-esteem, lower life satisfaction, a decreased sex drive, and menstrual cycle disruption. And going along with these, women are also susceptible to really developing a lot of the other symptoms that can be associated with these different problems. So going with depression, for example, women who develop depression as a result of a chronic yeast infection can also deal with insomnia, weight fluctuations, and just a lot of different social implications. And this is really an area of active research. There hasn't been a lot that's been conclusively stated yet about like chronic yeast infections and their play psychologically in a woman. But just from a lot of testimonials, women will say that it just kind of can bleed into a lot of areas in their lives.
So, how does it cause disease? So, the symptoms of a vaginal yeast infection can range from mild to moderate, but those main symptoms are itching and burning of the vagina and the vulva, a burning sensation, a redness or swelling of the vulva, pain and soreness, and then thick white vaginal discharge, which, as you can see to the right, is what that would typically look like. And so it's important to be able to distinguish what is like a different vaginal infection and what is a yeast infection. So some infections or diseases that a yeast infection may be misdiagnosed as is like a sexually transmitted infection, which is irritation, an itchy discharge, and a slight odor. A skin reaction or allergy, which you can just get from soaps or any type of like feminine hygiene product that is scented. Hemorrhoids, which is an itching or irritation in the vaginal area. And then lastly, bacterial vaginosis, which is an overgrowth of bacteria, which also has that discharge, and then an odor to it. So what sets a vaginal yeast infection apart from those is that it does have that fungus Candida albicans, and then it doesn't have an odor and there would be soreness. But it's important to be able to figure out that it's a yeast infection because it is fungal, which means it's a different treatment than something like bacterial vaginosis that you would treat with like antibiotics. You would want to treat antifungals with an antifungal medication. So the part of the body that a vaginal yeast infection affects is the vagina and the vulva, which are the tissues at the opening of your vagina. But also yeast infections can affect the penis, the skin folds, mouth, or nail beds. And then the fungus Candida albicans is responsible for most vaginal yeast infections. And then this happens to the body by this balance of yeast and candida and bacteria. And when there's an overgrowth of that yeast, that's what causes the symptoms of a vaginal yeast infection. And then also a penetration of the fungus into deeper vaginal cell layers can also cause symptoms. So some prevention, some ways to avoid a vaginal yeast infection is to avoid tight-fitting clothing, scented feminine products like bubble baths, tampons, and pads, hot tubs and very hot baths, unnecessary antibiotic use. We know from this class that antibiotics can harm some of those things that we need to be able to fight off things like yeast infections. And then staying in wet clothes, try to avoid that as well because the wet clothes can cause like a moist area that that fungus likes to grow on. And then what you can do is practice good hygiene, keep yourself dry and clean, relax and reduce stress because fungus infections can also come from stress, and then use antibiotics only when you have to. So some treatments for antifungals would be like the azole class of antifungals. So that would be like miconazole, clotrimazole, tioconazole, butaconazole, and taraconazole. But there's also other treatments like a short course vaginal therapy, which is taking an antifungal medication for 3 to 7 days, which clears it up pretty quick. A single dose oral medication is a one-time kind of stronger antifungal that gets it gone fast. And then a long course vaginal therapy is taking an antifungal over a long period of time. So that would be for like a patient who it's not going away easily on its own. And then the azole resistant therapy is boric acid. It's a capsule that's inserted in the vagina, and it's not taken orally, because if it is taken orally, it can be fatal. And then that would be for patients who may be like allergic to the oral antifungals.
So how effective are these treatments against vaginal yeast infections? In a standard uncomplicated case, your average topical or oral treatment will typically be effective within 3 to 7 days, and a woman can go back to living her life as normal. These treatments can really lower in effectivity in patients who have suppressed immune systems, just because it typically takes longer for the infection to clear up because of their suppressed immune system. And additionally, the effectivity can be really driven down in patients who are infected with a strain of Candida other than Candida albicans. And that's largely because those different strains can be resistant to the typical azole drugs, meaning that they'll have to go through a few different rounds of trial with azole drugs until they realize that the strain that they're infected with is resistant to that, and then have to seek another treatment, which can really draw out the infection. Which actually leads into an exciting potential new treatment. There is a new drug called ibrexafungerp, and it's being developed as a potential treatment against yeast infections, because it is a glucan synthase inhibitor, which is a major cell wall component in the fungus. And what's different about ibrexafungerp, just compared to like some of the azole class drugs, is that it has fungicidal properties, meaning that it's actively going in and trying to kill the fungus, where fluconazole and the different azole class drugs only have fungostatic properties, meaning that they're trying to really slow the spread of the infection. And what's really great about this is that it could be effective against a variety of Candida strands, not just those that are susceptible to the azole class drugs. And this could really benefit patients who are suffering from chronic yeast infections, specifically those caused by the resistant strands. And in lieu of other new treatments like a vaccination or anything, because that's not necessarily applicable here, many physicians are really advocating for improved education in the prevention of yeast infections, just because there can be some stigma and uncomfortability in addressing this when a woman is experiencing those symptoms. And so really trying to talk to their patients and just improve education, especially in the regions that I had mentioned before, where Candida albicans can really thrive. So tropical regions with poor sanitation education, and just helping women with preventative measures so that they don't have to experience a yeast infection in the first place.
So our questions are, the fungus blank is responsible for most vaginal yeast infections, so that would be Candida albicans. And then why does an overuse of antibiotics make a woman more susceptible to vaginal yeast infections? And then that answer is because an overuse of antibiotics can kill off the beneficial bacteria in the vagina, allowing for an overgrowth of Candida albicans. And here are our sources, and that is our presentation.
All right, good job, ladies. Questions for Amanda and Glory?
Yeah, I have a question. You guys might have said this, but what does Candida albicans do for women when it's in healthy amounts? Like, why do we have it?
Yeah, we actually didn't address that yet, but it's a lot of just a part of the vagina's natural microbiome, kind of helping to ward off different potential infections that could be coming from other sources, and just making it like one of the players in making it just the good environment that women need to be healthy.
Did you guys see anything, just out of curiosity, um, with some of the symptoms? Has there been anything that's looked at like decreased fertility in women that have maybe like the reoccurring, or the yeast infections that they can't get rid of? I don't know if that's a thing. I was just, when you're going over some of the symptoms, I was just kind of curious if that is a potential effect.
Yeah, I did not read anything about that, but I would think that yeast infections, like having them over a long period of time, that there would be some fertility problems, but I'm not 100% sure.
One more question for our team.
How long does the stuff last? Like, if you take treatment, like, how long do you have to take the treatment for? Like, do you know how long, like, you have to take treatment for it to totally go away?
Mhm. I think it just depends on how serious the yeast infection is. I think some can be, they can go away pretty fast, especially how depends on how your body reacts to the medication. But most, I think, can last. Some can last from 1 week to 2 weeks, but then some can last for months. So
Thanks.
Also, I was wondering, so you're saying that pregnant women can get yeast infections. So when they're pregnant with the baby, if they're taking medication, like was there any like effects on the baby, or like did they have the yeast infection longer because they were pregnant, or?
Yeah, so from what I was seeing, physicians who were treating their pregnant patients with yeast infections were typically leaning more towards topical treatments. I'm not sure if there would be any kind of adverse effects from taking an oral treatment, but I did see that that's something that they were leaning more towards.
All right, good job, Amanda and Glory. And next up, we have who do we have? Or is it dealer's choice?
We can go.
All right, Sarah and Lauren, let's do it.
Lauren, do you have it pulled up?
Yeah, how do I share my screen? Oh, I found it. Okay, one sec. Wow. There we go. Okay. Present. All right.
All right. So Lauren and I decided to research and present on C. diff. So I'll just start off with the epidemiology. It's known as C. diff pretty widely, but the actual name is Clostridium difficile. It's also known as Clostridioides difficile. Yeah. So it's a bacterium found in intestines, so in your gut, that can cause a lot of health issues, and it ranges from frequent diarrhea to life-threatening colitis or colon inflammation. And so if you look on the left, you can see that it is a gram-positive bacilli bacteria. And on the right, that is what your gut looks like when you have it. All right, so a little bit of history. There's a few important dates that go into C. diff. So it was initially discovered in 1935, but it wasn't actually until 1978 that people found out that it had toxic properties and could cause all of these health issues. So in 1935 in Denver, Colorado, it was isolated from stool of a healthy infant by scientists Hall and O'Toole. They were basically just doing some different cultures, looking at infants before 10 days old, and they found C. diff, but they didn't find any problems. There was no diarrhea or any other symptoms that showed up, so they weren't aware that it was linked with those issues. They did, however, find that it was toxic to animals, and they saw the toxic properties, but they didn't connect that with humans for quite a while. So then in 1978, George and Bartlett recognized it as a cause of antibiotic-associated illness. And this basically was linked with the toxicity in rodents, and they were able to link that with an uptick of diarrhea and abdominal pain in hospital settings. And they connected that with the use of antibiotics, and I'll talk about that in just a little bit. But in pretty recent history, in 2001, it was really more between like 1999 and 2004, so like the turn of the century basically, there was an emergence of a new, more drug-resistant and more toxic strain of C. diff. And to give you guys an idea of the scope of that, basically in 1999, there was about 5.7 deaths per million. And then in 2004, that changed from 5.7 to about 23.7. So it's a lot more toxic, and it's also causing issues outside of the hospital too nowadays. So the spread of C. diff. C. diff spores are what are most dangerous about it, because those are very difficult to destroy. And they're shed into feces of infected people. And then when people don't effectively wash their hands, it can get spread to basically anything that they touch. So it can be spread to food, and then when you eat the food, you get it. It can be spread to surfaces, and then when you touch things. But the most common way that it spreads is through human contact. So handshakes or just touching another person. People can also carry C. diff in their gut, but not become sick and not show symptoms. However, there still are some rare cases where they do spread that infection to other people. And I'll talk about that in just a little bit too. It's most commonly spread in hospital environments and other long-term care facilities. That's about 70% of cases. This is mainly because of the antibiotic use in those environments, but also because there's a lot of bacteria. There's a lot of potential for it to spread because there's a lot of immunocompromised and vulnerable people, and they're all very close together. It's commonly spread, like I said, hand-to-hand, human contact, but also on things like cart handles, phones, bedside tables, toilets, sink faucets, stethoscopes, basically anything that you touch and another person can touch. And it's not on the slide. Another common way that it is spread is actually through pets. So if you come into contact with the fecal matter of a pet, like if you're walking your dog in the park and you got to pick up its poop and it happens to have those spores, that's another way it can spread. So there are quite a few effects on life outside of health, and Lauren's going to talk about the symptoms a little bit later. But there's plenty of journal articles and studies that show that C. diff has a lot of negative effects on the impact of your quality of life. And basically that's just because if you think about it, if you have frequent diarrhea and intense abdominal pain, it's going to change the way that you live your life. So that affects all of these things in the list that you can see: your self-care, your usual activities. So whether that's your hobbies or just your general mobility, it'll heighten pain and discomfort, and that can lead to anxiety and depression sometimes because of the huge change in lifestyle. And CDC recommendations are that you don't go to work or school while you have symptoms. So you can be out of work and out of school and fall behind in those things until the symptoms go away. And Lauren will talk about that in a couple slides.
Okay, so the causes are, well, basically it's just one cause. Typically it's an infection that you get because your normal microbiome is altered or eliminated through antibiotics. And like she said, their spores from C. difficile are in feces and can spread to food or surfaces when individuals who are infected don't wash their hands thoroughly. These spores can live for weeks or months on food and surfaces, so they'll be there for a while if it's a contaminated surface. And when you touch one of these surfaces or you eat the food, you can ingest the bacteria, and then it will colonize in your colon. And the majority of infections occur in people who have been in a hospital or a long-term care facility because, like she said, there's more antibiotics and more chance of getting contaminated. So the pathology is that your normal protective microbiota is eliminated or suppressed, normally by antibiotics, and then you either ingest C. difficile or the overgrowth of it occurs in your colon. And so some individuals naturally have C. diff or C. difficile in their microbiome. And so when their normal microbiota is altered, then they're just going to have an overgrowth of C. difficile, but other people don't have it, so they have to actually ingest the spores to get sick. And then the spores germinate in your colon, and they grow toxin-producing cells on the mucosal layer of the large intestine and colon. And what causes the inflammation is that toxins are released, and the mucosa becomes inflamed. So you typically get this 5 to 10 days after antibiotic use. That's how long it takes for the spores to germinate and the cells to grow, and then for those toxins to be released and cause inflammation. And so if you have a mild or moderate infection, you're going to have abdominal cramping and malaise, which is generalized weakness, and diarrhea 3 or more times a day for 2 or more days. But if you have a severe infection, you're going to have diarrhea like 10 to 15 times a day. And you're going to have a fever, and you're going to get blood in your stool most likely because the aggravation of that mucosal lining is going to cause it to bleed and become raw. And then you're going to be dehydrated from all your diarrhea. And if dehydration occurs quickly, you're going to have an electrolyte imbalance, which can cause your kidneys to fail. And then you're going to have colitis, which is inflammation of the colon, weight loss, and you can have an increased white blood cell count because you have that infection and your body's trying to fight it off. And you can have a swollen abdomen due to your kidney failure. It can't filter out that fluid, and sepsis, which is when the bacteria gets into your bloodstream, and that is what causes death. So for your initial infection, you're most likely going to get vancomycin as an antibiotic, and they're effective in keeping C. diff from growing, but 20% of people have a recurrent infection, either because their first strain wasn't taken care of or they get a new strain later. So vancomycin is going to inhibit the peptidoglycan, so that's how it's going to kill the bacteria. Fidaxomicin is relatively new, and it is specific to gram-positive anaerobic bacteria, which is what C. diff is. So it doesn't alter your natural microbiota as much. It targets RNA polymerase. So that's how it causes that. And then metronidazole, which we talked about in class. So another treatment is going to be fluids with electrolytes to replace all that you lost from diarrhea and to prevent kidney failure. And then some people actually have to have surgery, which removes the disease portion of the colon or your affected organ. So if you have organ failure or toxic megacolon, which is when your colon becomes distended and swollen, and so it can't get out all of the gas and feces, and it can actually rupture. Or if you have a perforated bowel, which from those toxins, it can create a hole in your bowel, which then would leak your feces into your abdominal cavity, and that is really bad and will make you really sick. So you have to get surgery for those things. And if you have a recurrent infection, they're typically going to use a different antibiotic course than they use for the first infection. So whichever one they use the first time, they would pick a different one, because the second time that you use that antibiotic, it's not going to be as effective. And then there is C. diff that is resistant to antibiotics. So that is the bad stuff. So to treat that, they've been coming up with these future treatments, like we've talked about in class, fecal microbiota transplant, which is when you take the healthy microbiota of someone and put it into the diseased person. They've done this some in clinical settings, but most of them are still in studies, and it's shown that it has a 94% cure rate versus a 31% cure rate with vancomycin bowel lavage, which is when they like rinse out your bowels with vancomycin. So it's proven to be a lot more effective in some studies than that. And in recurrent patients or refractant patients, bowel movements were normalized in a week, which is really good. So they've also been looking into probiotics, and probiotics that are available right now haven't really been shown to be helpful for treatment or prevention. They're helpful to restore your natural microbiota after you've taken your antibiotics to get rid of C. diff, but as far as like actually getting rid of C. diff, it hasn't done anything effective. But they're looking into new probiotics that can actually secrete antibiotics in your gut, and so they're looking into that for future treatment. They have been looking into vaccines as well. So one of them that they've created is a toxoid vaccine. So it is administered against the actual toxins that C. difficile produces, or recombinant vaccine that administers DNA that codes for the toxin, so your body can mount an immune response to those toxins, and if you actually get C. diff, then they won't affect you as much. And passive immunotherapy, which is when they take antibodies to toxins A and B and inject them into the patients. And so these didn't really help with initial infections, but they reduce recurrences by 72%, which is really good. But they're really expensive, so they're going to have to work on something to bring that price down before it can be used in everyday treatments. And they're also working on different antibiotics. And so this one is a diazolid, which is a broad spectrum antibiotic. And so the problem is that it does kill all of your good microbiota as well as C. diff, but it inhibits anaerobic metabolism, so that's a little bit more specific to C. diff. And the problem with this one also is that it's really expensive, but it can be used for recurrent cases instead of initial cases, so it wouldn't have to be used as often. And so that could just be another treatment if it goes through. So these are our questions, and it was, how does C. difficile get the opportunity to colonize the colon of humans? And that's that the normal microbiota is suppressed or eliminated by antibiotics. And what age group is most likely to become sick from C. difficile? And that's going to be 65 years and older, because they don't have as good of an immune system. And these are our sources. That's it.
All right, any questions for Lauren and Sarah? Questions? Come on.
Did any of your articles talk about the amount of patients whose infection like progressed to be severe or required surgery?
Not really. They didn't talk about amount as much as just like ways to treat it. But I think recurrent infections are pretty high, but as far as like how severe they become, it didn't really say.
Yeah, I had some information that said for patients over the age of 65, about 1 in 11 of those people died within a month of diagnosis of C. diff.
All right. One more question. Come on, one more question. I believe. Come on. We can sit here. I'm feeling pretty relaxed.
I think you guys said something early on about they originally got it from fecal of a newborn. Is that right?
Yeah, that's right.
So was the newborn infected, or like how did it have it?
So basically, quite a few people actually carry the C. diff bacteria in their gut, but there's like the good bacteria that suppresses it, which is why when people take antibiotics, they target the pathogens, they also kind of take out some of the healthy bacteria as well. And that's when C. diff can get out of control and the symptoms start to show. If that answers your question.
Okay, cool. Anybody else? I like it. All right. So here's what we're going to do. There's been a little hiccup in our plans. I've lost Anna, who is supposed to present today. So what we're going to do is we're going to take a break until 9:30. I'm going to lecture a little bit more, and then we'll just add that on tomorrow for our presentation extravaganza. All right? Cool. Get excited. Let's take a short break. And if Anna somehow reappears during that break, I still will probably let Anna and Olivia go. But if not, you'll get to hear me talk, which I know you guys are super stoked about. All right. Take a short break. Good job on all the presenters today thus far. They've been really good. Zoom recording. So remember we were talking about HIV, and we had specifically right before this talked about MMWRs, right? Those morbidity, mortality, weekly reports that physicians and other clinicians, right, can publish and send into the CDC and allow us to track infections that we are concerned about, and also allow us to identify new infections. So this is the HIV virus. Notice at least the external structure and some components of the internal structure are somewhat similar to coronavirus, right? You have these nice little spikes, right, that give the coronavirus its name. A similar structure, right? Different protein components, different size, but still similar. It's also an enveloped virus, just like the SARS-CoV-2. There's a couple important things that I want to point out on here. All right, one of those being, or kind of the ones that we'll focus on the most, or most important in my opinion, are surface glycoprotein GP120 and surface glycoprotein GP41. These make up the spike, all right, and are essential for attachment to the CD4 and then the CCR5 or CXR4 receptor, right? Specifically GP120 there, and then GP41 is going to be an essential part of the spike that's going to allow entry into the target cell, all right? You also see down here caspid protein P24. It's the most abundant protein that's present in capsid of HIV. And this is actually one of the things that we look for in blood to diagnose or confirm an HIV infection, is the presence of this caspid protein. All right. You notice here HIV is an RNA virus. We'll talk about that a little bit more. We also have some really kind of unique enzymes, specifically reverse transcriptase and integrase. And we'll talk about why those are kind of unique for this virus. So that's kind of what's going on that picture. And so here's just showing you HIV entry into a cell. And here you see the spike, right? GP 120, right? The outer portion of that spike connecting to CD4, right? We then, from there, proceed to CXCR4 or CCR5, right? CCR5 is going to be the most common co-receptor that the spike attaches to. And then, right, GP41 is going to initiate or allow entry, right, membrane fusion that puts the caspid into the cell. Okay. So again, there's nothing overly unique here that we haven't discussed before. You guys kind of understand, or hopefully remember from chapter 13, kind of the basic mechanism of viral entry into a cell. All right. So here's where we kind of point out some really cool, interesting things about the HIV virus. All right. So, right, the caspid along with all those different enzymes and nucleic acid, in this case RNA, enters the cell. And here's where we see something really interesting. All right. So DNA, right, is made from RNA, and this is done with reverse transcriptase, right? So usually we go DNA to RNA to protein. We're not doing that here. We're actually going RNA back to DNA, right? And again, we make one strand, so we make a complementary strand to RNA. We then, right, have our anti-parallel strand of DNA added to that. And so now you have double-stranded DNA, just like we normally have in our cells, all right? And what's really cool is that this HIV DNA takes a circular form, right? Almost kind of like a bacterial plasmid. And then we have two options, right? One, right, is this DNA can be turned back into RNA, right, for a new virus that's going to be packaged. Or what makes HIV really interesting, right, is that it can integrate itself, right, into a host chromosome. You remember we talked about bacteriophages integrating themselves into their host cell, right? We would have a prophage. Well, here, when HIV integrates into the host chromosome, we have a provirus, all right? And then later on, right, it can leave the host chromosome, right? And so this is very similar in some ways, right, to the infection cycle capabilities of a temperate phage. All right, this kind of reverse transcriptase, right, is unique, all right, and then in animal viruses, right, the enzyme integrase is also pretty unique, right? So this gives the virus a great place to hide. Right, and then you can see that other than that, though, right, viral replication assembly is similar to what you guys already know, hopefully from chapter 13. All right. So here's one of the things that makes AIDS, or in this case, right, the HIV virus, pretty cool, right? So this little sun here represents a individual HIV viron, right? This is actually a dendritic cell. And what the virus has gotten great at is essentially hitching a ride with the dendritic cell. So the dendritic cell, right, it likes to sample things. It samples, right, it finds some virus, it grabs onto it, right, it interacts, right, like we talked about before, antigen presentation with a T cell. And what you've essentially done now is you've dropped off the virus at its preferred target, all right? Once in its preferred target, right, it then does what viruses do. Now, this is what's really cool, and again, cool, not good, but I think cool from a biological perspective with the HIV virus, is that you noticed, hopefully a couple slides ago, right, or remember when I said, hey, it's a provirus, it integrates itself into the host chromosome. Well, where does it integrate itself? It happens to be that it integrates itself just downstream of an NF-kappa B binding site, all right? Does anyone remember the role that NF-kappa B plays in cell activation, inflammation, all of that fun stuff?
It's a natural killer cell. Like it kills like pathogens and things like that.
Say that again to me.
it's like a natural killer cell. Like, so it kills like pathogens and things like that.
Well, so natural killer cells, right, kill pathogen infected cells, but what does NF-kappa B do in inflammation and communication amongst cells in the immune system? What is it for?
Doesn't it-
Yeah.
Oh, cytokines are like produced from the genes that are transcribed and translated.
Perfect. So like NF-kappa B is the main transcription factor that is leading to communication, that is leading to activation, right? It is essential. And so this is, I would say, pretty devious, right, by the HIV virus, because it has inserted itself downstream, right, of the NF-kappa B binding site. And whenever an immune cell is activated or stimulated, right, which is often, right, whenever it's doing its job, that means not only are the genes going to be copied that are for those cytokines that we need, but that means the HIV virus is copied every time that cell is activated or trying to make copies of those essential cytokines. All right, and so, I mean, it's an ingenious kind of place to hide. You hide downstream of the binding site of the transcription factor that is for genes that are an essential part of your function, right? Every time an immune cell does its job, right, we're getting another copy of the virus, right? So then eventually it just starts to multiply and multiply within that cell. And it will, at some point, lead to dysfunction and destruction of that cell. All right. Anyway, so what is AIDS specifically? All right, we've talked about this is kind of the end result of a prolonged HIV infection when we start to succumb to rare infections or infections that normally would not cause any type of pathology in someone with a normal immune system. So there are two things, all right? You need, right, a CD4 T cell count. Okay, well, let me tell you. So there's two things. And one of the first things is going to be presence of the virus, right? So they have to be able to find or detect the virus in your system somehow. They then need one of two things, right? This or this, right? So HIV virus, right, in your system that they can detect, and, right, a CD4 T cell count of less than 200 cells per mil of blood, right? Or HIV, right, detection, and one of these defining kind of diseases, right? One of these rare diseases. So if you have the virus in your system and you have one of these infections, boom, that indicates that you have reached the point of AIDS. If, right, you have the virus in your system and this super low T cell count, then we would say, hey, you have AIDS, all right? So you need the presence of the virus and then one of these two possibilities saying that, okay, we've reached the point of having AIDS. All right? So again, you know, the complications are things that normally, right, we would be able to deal with. So Kaposi sarcoma, right? That in normal individuals is a very, very slowly developing cancer that does not affect quality of life or length of life. But again, in these HIV patients, because of their suppressed immune system, it moves quickly and it shows up at a much younger age than we typically see, right? Pneumonia and respiratory infections in general are huge issues for HIV infections, right? And so, right, yeast infections that Amanda and Glory were talking about, right, from Candida albicans, can actually become deadly in HIV patients, right, because that fungus can move to different areas of the body that it normally wouldn't infect. Cyclomegalovirus, if any of you have had mono, this is one of the two ways that mononucleosis can be caused. And so, you know, normally, if you have a healthy immune system, you have mono, you recover from it. For the most part, you're fine. Not the case here. neurologic and renal complications, right? If you remember, HIV can infect neurons, and so we sometimes see that in patients, and then they can have kidney issues too. But again, all of this in someone with a normal immune system would typically be relatively easily dealt with, or at least would be resolved after treatment with medication, right? That's not the case with HIV patients. So, they are producing 10 billion, right, new individual viral particles per day, right? So there's a lot of the HIV virus in their system. And so one of the main things that we use to treat HIV is something called HAART, highly active antiretroviral therapy. And so, I'd have to look. That used to be these patients would be taking a pill for each one of these different things, perhaps multiple times a day. I'm assuming now that they've probably combined some of these things, so it's a little bit less medication. But basically, within the HAART treatment, you are doing multiple medications that are interrupting multiple cycles or multiple sequences or steps within viral infection. So here, right, part of the HAART treatment, you're going to try to block these entry inhibitors. So you're going to block CCR5, and you're going to block CCR4. So without that co-molecule to help dock it to the cell, the virus can't get into the cell, all right? NRTI and NTRTI, right? So this is competitive inhibition of reverse transcriptase, all right? So reverse transcriptase, if you remember, is taking that viral RNA and turning it into DNA. And so if you prevent that from happening, then the virus can't make copies of itself and can't integrate into the host genome, right? You also have non-competitive inhibition of reverse transcriptase. And so if you guys go remember your enzyme basics, you can either block the active site of an enzyme, or you can bind to something called an allosteric site, which changes the active site of the enzyme. Either way, right, it can't bind its substrate, and then it can't function, all right? So in this case, we're preventing it from binding to RNA to make that DNA copy. Integrase inhibitors, if you remember, integrase is essential for incorporating the incorporating the viral DNA into the host chromosome. And so, preventing that from happening interrupts the infection cycle. And then finally, protease inhibitors, not necessarily unique, but protease inhibitors are going to interrupt assembly and final maturation of that virus, right? And we've seen over 70% decrease in AIDS and mortality in HIV positive patients. And so most Western countries that have decent healthcare have HAART treatment available, at least a lot of places in the United States. Even if you can't afford it, the government will provide the treatment to you, right? This was not always the case. I haven't watched it, but the movie Dallas Buyers Club, right? At certain points in our history, these medications were not available to all. It were super, super expensive. And so that movie kind of focuses on that a little bit. But now this medication is readily available for anyone that needs it here in the United States, right? Not the case in poorer countries throughout the world that are still dealing with severity of the HIV pandemic, right? But here we can treat it pretty well. Can anyone think of a negative consequence of the HAART treatment from a societal point of view? None. Professor Perry, can you think of a negative consequence of the HAART treatment or the effectiveness of the HAART treatment from a societal point of view?
I'm not sure what you mean by societal.
Yeah, that's okay. Maybe I'm not wording it correctly. Has anyone heard about the AIDS orphans in Africa recently? No. Has anyone been to a AIDS concert recently or done an AIDS walk recently? No. So before the HAART treatment, when HIV was killing large numbers of people, right, it was an incredibly active area of research and drug development. There was a lot of effort and work put in from the public health side, educating people how to prevent the spread of HIV. Has anyone seen any commercials on TV recently about HIV medications?
I've seen like the PrEP ads.
Okay, and what are they saying the treatments for?
Preventing the initial infection.
Yes, right. So, you know, one of the main ways that HIV is spread is through sexual intercourse, right? And so now, right, we have commercials for medications that are saying, hey, if you're sexually active and you have HIV, right, go ahead and take this pill and help prevent the spread of it, right? And so it's almost a little bit of a 180, I think, from our initial concern, you know, for the spread of the virus. And, you know, part of the problem is the HAART treatment is so effective that most of these patients that are getting treated have relatively normal life expectancies and even normal quality of life. And because of that, it's not as big in the public eye as it used to be, but it's continuing to spread. We don't have a cure and we don't have a vaccine. And so I'm not saying, I think the HAART treatment's incredible, right? What it does for these patients' quality of life is incredible. But in some cases, from a research medical standpoint, we've almost become, I think, a little bit of a victim of our own success. And, you know, there are some scientists, now again, they're not in the majority, but, and I don't know, I can't remember what the timeline is, but predict, you know, sometime way, way, way in the future, you know, everybody might be positive for HIV, and we will all in the future take, you know, like one pill that kind of suppresses the virus, right? And that's just, I think, the fact that someone's even making that prediction, I think, has shown a little bit of the complacency that we have towards this. Now, I say that too, there still are labs that are doing HIV research and are really interested in trying to solve this problem, but it's just not to the scale that it was before. And I just kind of put this out there as a little bit of a warning. You know, sometimes pandemics, unfortunately, I think become a little bit like the flavor of the week, right? I guarantee you, sometime within the next 5 years, maybe even sooner, hopefully not though, because I'm ready to be back in the classroom and not teaching remotely again. But there might be, you know, something else, right? Remember Toomey's article, a few years ago, it was Zika. And now we have COVID-19, which is super, super serious, right? A few years ago, we had Ebola, right? And so there's always something, and at least initially, right, the research on that is really, really intense. And then sometimes it unfortunately dies down a little bit. And HIV, I think, unfortunately, is an example of this.
Hey, Dr. Daft.
Hey, Mercy.
I have a question. So, did HAART treatment replaced ARVs?
Antivirals?
Yes.
Antiretrovirals?
Yes.
I think those are part of the HAART treatment. Or they're on it together. I would have to check, Mercy, and that's a great question. I haven't checked to see what the modern kind of, Professor Perry, do you know the modern kind of medication regimen that an HIV patient takes?
No, I don't know the exact drugs, if that's what you're asking.
So Mercy, that's a great question. I'll look up and see kind of what a typically daily medication list or dosing is for HIV patients, because that is an important question.
Okay.
So one of the things that I want to talk about HIV that is incredibly fascinating, right, is a mutation in the CCR5, right, that co-receptor that the virus uses for entry into the cell. And so there's something called a CCR5 delta 32, right? So that is a 32 base pair deletion from the CCR5 protein, right, receptor. And this results in a non-functional protein. And we actually see this in about 10% of Caucasian Europeans and those of Caucasian European descent. This same mutation is incredibly rare in Africa and Asian populations, right? Interestingly enough, this is one of the things that I find fascinating, and I have a paper on this that before COVID-19 stuff started happening, I was going to make you guys read it, but the students of the fall might be my victims for that. But the same mutation, the Delta 32 of CCR5, actually protects against smallpox infection. One thing I want to kind of point out, and you see it here, is that although it's protecting from HIV and smallpox, right, this is not necessarily something that you want. There is impaired T cell function, and you do have an increased susceptibility to the West Nile virus. So, you know, if the African community, right, whether that be African American, right, or actually, right, living in the continent of Africa, why does this jump out to you when I talk about this mutation and I say that it's rare here? Please don't overthink it.
Like natural selection?
Natural selection in a sense, yes, I can work with that, right? If we look at the prevalence of HIV, all right, not the total numbers, but the percentage of population infected, right, we see the highest rates in sub-Saharan Africa. And one of the reasons, not the only reason, but one of the reasons that we see this is because there is no naturally built immunity in these populations, right? And interestingly enough, not that Africa or Asia were spared from smallpox, but way back when, right, smallpox was a huge problem in Europe. And so kind of to Megan's point of natural selection, what we see is that way back in the day in Europe, when smallpox rampaged through the continent, right, a number of the survivors, right, likely had this mutation. And then what do survivors do after pandemics? Rachel, D.
They pass it on?
That's right. They fall in love and they get married and they have babies, right? And that's all the detail I'm going to go in. If you want more detail, right, take A&P2 with me and we can cover that, right? But that is the general idea, right? They survived, right? Smallpox pandemic. Maybe they had the mutation, right? They got together with other people that had the mutation. And that's why, right, generations later, we see a little bit of naturally built-in protection from HIV infection. Okay. So it's I don't know, I think it's a really interesting kind of, you know, piece of genetic history that we can trace and look at. Now, for HIV, what's really interesting too is we have a group of people called elite controllers. And these elite controllers have an HIV infection, right? We can pull it out of their blood, but they maintain a normal CD4 T cell count, and they have a much lower viral burden than we see in the majority of HIV patients, right? So this is really, really rare, right? And what we've noticed, or what we know about these patients, is that there seems to be a genetic component. So this gene FUT2, which is associated with ABH blood types, is present in these patients. They have similar MHC, so that's HLA, it's another fancy way for saying MHC, similar MHC receptors. We notice that they have a unique antibody profile, and they seem to all have the A3 gene, which naturally inhibits reverse transcriptase. All right. I'll be honest, I need to look more into these patients to see if they have any more insight into how these are protective. So this is, in a sense, good for a patient that has HIV, right? So it means they will probably live a normal life. Why is this bad for everybody else? And again, please don't overthink this. Gloria, why is this bad? Why is it why is it kind of worrisome that there are elite controllers within the population?
I'm not sure.
What's one of the big concerns about the spread of COVID-19?
Well, they could be like a risk to everybody else.
Right. So perfect. Right. So for COVID-19, right, one of the big things is that a lot of people, that have that virus infection appear to be asymptomatic, right, showing no symptoms or showing very mild, symptoms. For the most part, right, those SARS, CoV viruses or corona-like viruses are only causing like mild colds, all right? And that is the case for COVID-19, and that's what's kind of dangerous. You either don't show symptoms, or you maybe have a little bit of a sniffy nose and a little bit of a cough, but nothing major, and so you don't think anything of it, right? And then you spread it. So that's what we're really concerned about. And elite controllers would be a population that we potentially would be concerned about, because if they're going to the doctor and their CD4 T cell count is getting measured, they can do that when they draw your blood, right? It's really simple, and it always looks normal. They might not suspect unless it becomes a case of self-reporting, right? So maybe their partner comes down with HIV and then they go and get tested. But unless there's some type of self-reporting or self-assessment, these patients could go unnoticed and continue to spread the virus. Okay. And so, one of the things you guys have been pointing out today in your 3 presentations is, what does this look like today in society at large? And I'm sure Professor Perry remembers this relatively well, right? Magic Johnson became kind of one of the poster, we'll say, children of HIV, and this was a big deal, right? First of all, I told you that it was often associated with homosexual men. And even though before this we knew that wasn't always the case, right? Getting people to kind of change their minds about that was tough. Right or wrong, it was, seen as a disease that probably only affected a certain socioeconomic class. And so Magic Johnson, it was a big deal, right? I mean, he was one of the best players in the NBA, right? We could go into it, right? He's the best point guard ever. And I dare any of you to, debate me on that later. But he was a really prominent figure, right? The Lakers had won multiple NBA championships. I mean, he's at the top of his game, just super good. He also brought to light kind of societal fears on how HIV is spread. So one of the reasons that he retired, or his first retirement, was because players were afraid to play with him, right? We didn't understand the virus fully. People thought if he sweated on them, right, that they would get HIV. People were worried, well, what happens if he gets a small cut and he bleeds on me, right? Will I get HIV? And so it really kind of, I think, forced society to take a deeper look, at this. And he did come out of retirement again briefly for a few years and played even with HIV. And some players, right, reacted differently. And now, again, I'm not sure how many athletes have HIV, but it certainly is not looked at the same way as it was before, in a good way, right? And so Magic Johnson was kind of a key figure in pushing that discussion forward. And if any of you watch basketball or TNT, or your parents watch it, right, Magic Johnson is still a very prominent and vocal figure in the NBA and is doing quite well, right? And so he was diagnosed in 91. And so what are we going on? 30 years now since he's been diagnosed, and over 25 years that he's been on heart treatment, right? And so this is one of the things, right, that I was pointing out, that kind of normal life expectancy that we see in some of these patients, right? All right, and so this was this is kind of a sad story, but this is Stephen Crone. He was one of the gentlemen that allowed us to identify and start to understand the CCR5 Delta 32 mutation. And so Stephen Crone was homosexual, and he had homosexual partners in the 70s and I believe 80s. And they started dying later on of HIV. And he did not go in and get tested because he just assumed that the partners I had were dying from the virus, and so that he would also eventually die. Time continued to go on. He did not die, and he went in and got tested, and he's one of the gentlemen that they found this mutation on, found this mutation in early on. Unfortunately, in 2013, you can see here, is when he passed away. He committed suicide. A lot of it related to, the number of people that he had known that had died of HIV. Right. So important, though, in our understanding of this mutation. All right. So here's our one gentleman that appears to have been truly cured of HIV. Right. So his name is Timothy Ray Brown. He's called the Berlin patient. He had leukemia, right? And he, for his leukemia, received a bone marrow transplant. And it happens to be that his match for bone marrow was a person that had the Delta 32 CCR5 mutation. Okay. And so he basically, right, what a bone marrow transplant is basically giving a person a brand new immune system. And so the immune system that he received actually, right, was resistant to HIV infection. This, from a scientific research standpoint, is completely fascinating. From a medical treatment standpoint, it is as impractical as it gets, right? This is not a cure for the common patient. Those of you that have heard anything about bone marrow transplants, finding a bone marrow match is incredibly rare, right? I'm not one for numbers, but I can tell you finding a bone marrow match and then finding a bone marrow match with a Delta 32 CCR5 mutation is relatively astronomical, right? This is like winning the biggest lottery ever in the world, right? Not something that can be used on a wide scale, right? Let's see, this is the last one. Yeah, the last one we're going to talk about here, and then I'll break you into your lab groups, is the Mississippi baby. This was a baby that was born with HIV and was immediately put on a heart treatment. And they monitored this baby for a couple of years, and it appeared that the baby was cured. All right, so they were super excited. They could not detect any part of the virus in her blood. And then her mom unexpectedly moved away with her, did not tell the doctors. They lost track of her for around a year. She came back to the same area of Mississippi a while later, went back into the doctors, and the doctors took the little girl's blood, and she had HIV, right? They could detect it. So they immediately put her back on the heart treatment. And so this kind of relates back to Mercy's question a little bit. If a mom tests positive for HIV while the baby is in utero and she's not already on the heart treatment, they immediately put that mom on the heart treatment. And typically what we do now is those babies will also remain on the heart treatment. And this is where it becomes kind of interesting from an ethical standpoint, right? So if you put a baby on the heart treatment, what's the only way that you can test and see if it cured the baby? Caroline, what would you do? Have to test if the baby was cured?
You would have to test the mom.
Well, no, not the mom, right? So let's say the baby's born, right? And you have the baby on heart treatment. So how do you find out if the heart treatment works? How do you find out if the baby's cured? What do you have to take away?
I guess you have to take away like their parental say in like testing them and like giving them the treatment.
Yeah, let's say the mom signs off. She's totally fine with it. they would have to sign off on something. What would you take away from the baby to see if the heart treatment cured them of HIV?
You take blood?
You would take blood, right? Someone help me finish it off. What am I taking away?
The medication?
The medication ultimately is what you're going to end up taking away, right? Does anyone have any ethical issues about taking away a treatment that we know suppresses HIV and allows the patient to live a normal lifespan to see if the virus can become active and detectable in their blood? I hope for most of you the answer would be yes, although I understand from a research perspective the question is so tempting to know, right? But babies that are on heart treatment they remain on heart treatment for the rest of their life. Now, I guess presumably later in life, when they become adults, they could say, hey, I'm going to go off of it and let's see what happens. But from an ethical standpoint, right, if you know that it suppresses the virus, you know that you're not able to detect it in their system when they're on the treatment, why would you risk taking them off, right, and letting the virus multiply and become active in their systems, right? And so it's one of those kind of questions that we may not know until years down the road if some of these children, as they become adults, agree maybe in the future to go off the treatment to see if the virus comes back. But even then, I don't know if I was one of them. I don't know if I would want, to risk that. And so we know the heart treatment works, and so sometimes we just go for it. All right.
Okay, Dr. Deft.
Yes?
Hey, Dr. I have one final question.
All right. Professor Perry is ready to answer it.
Okay, so how possible is that? Because back home, I saw people who were like, both the parents were HIV positive, but they ended up giving birth to an HIV negative baby. How is that possible?
Woo! I'm glad I volunteered Professor Perry for that one. I have to look. I'm not sure if, and Professor Perry, if you do know, you can correct me on this. I'm not sure if the virus crosses the placental barrier in every situation. And what I don't know too is, if you test those kids in a couple of years, do they have detectable level of the virus? Professor Perry, do you know anything about the vertical transmission route?
Well, I don't think it's 100%. I think there is a chance the baby could be born without HIV, but I don't know the statistics or, what the chance. I doubt that I think it's much more likely that they would have it.
Right. And one of the things, right, with birth too, is even if it doesn't cross the placental barrier, if any of you ladies, and I know Mercy already has a kid, but if any of you other ladies decide to have, children, it's a relatively, I'll say physiologically taxing process. And it's not uncommon, especially right during natural births, for small vaginal tears and things like that to occur. And in that case, right, the mom starts bleeding and the baby's exposed to the blood even during birth potentially, right? And so I'll have to look, more into the stats, like Professor Perry was saying, because I got to look at the numbers, but that's one of the reasons that, if a mom's pregnant with HIV, they put her on the antiretroviral to increase the likelihood that it doesn't cross the placental barrier, right, to kind of to boost that chance. But I'd have to look into those numbers too. So yes, interesting stuff. I'm going to stop this recording.